Hello there, Year 9, and welcome to the last week, week 15 of science. This will obviously be the last week before the holidays, and I think it would be a good amount of time to go over the bioenergetics chapters of the course. I've split this into two, so today we'll do photosynthesis, which includes photosynthetic reaction, rate of photosynthesis, and the use of glucose from photosynthesis. Tomorrow we'll do respiration, so the aerobic and anaerobic respiration, response to exercise, and metabolism. Then there will be some exam style questions based on these two topics. And then the last two lessons will be going over those as a walking, talking mock. So I think this one out of the two that we did was the most beneficial, this method. So I'm going to be going through the sections of photosynthesis today, and you are able to do whatever method you want for your revision. So you can do it as a mind map, you can do it on revision cards, you can do it as notes, however you want to put the important information. I'll go through each slide, and then if you pause the slideshow, pause the video, then you can add the important parts to your revision notes, and then we'll move on. So we're going to start off with the actual reaction itself. What is photosynthesis? So if we break down the word photosynthesis, photo means light, like a photograph is where you have the light on a picture, and synthesis means to make. So photosynthesis is using light to make something, and what they're making is glucose, and that's how plants get their food. Unlike us, where we eat and take in glucose, plants make their own, and they're very important because it will be the basis of all of the food chains. Now, a plant manufactures glucose from carbon dioxide and water, and it uses the energy from light in the chloroplasts to combine those two together to make glucose. Because energy is going in, it is an endothermic reaction. So this links to the chemistry energy topics, which you'll be doing next year. There's two types of reaction, endothermic and exothermic. Exothermic gives off energy, endothermic takes in energy. So that is the reaction in terms of the chemistry of it. So you can pause the video now and put down any of the important parts. I would suggest paying particular attention to anything that seems new to you or that you didn't know beforehand. Okay, so we also need to know about the reaction, the word equation, and the symbol equation. Now, the word equation is straightforward. So you've got carbon dioxide and water. It doesn't matter which way around you have those. React to make using light. So we put light at the top of the arrow. Sometimes you will see chlorophyll underneath the arrow because it needs chlorophyll to do it as well. And it makes glucose and oxygen. Now, you're expected to be able to know or recognize the chemical symbols for all of those things. So we can put it into a symbol equation. Carbon dioxide has the symbol CO2. Water is H2O, and that makes glucose, which is C6H12O6, and oxygen, which is O2. Now, it's really easy to balance because it's all the same number. It's just 6x6, and that goes on everything apart from glucose. So if you could pause the video and make sure you've got the word and the symbol equation for photosynthesis in your revision map, notes, cards, any way that you are using. Okay, so now we're going to move on to the rate of photosynthesis. And if you see anything to do with rates, it's just how quickly something is happening. So it's over time. So carbon dioxide is one of the reactants that is needed to make glucose. The more carbon dioxide, or the higher the concentration of carbon dioxide, the faster the rate of photosynthesis. But this only happens up to a point. So you can see on the graph, which we need to be able to recognize, that as you increase the carbon dioxide concentration, the rate of photosynthesis also increases. And that happens to about 0.1%. And then it plateaus. It reaches its maximum point. Now, you don't need to know that the maximum point is 0.1%, but you do need to know the levels off at a certain point. And the reason for that is because there's other things that are limiting the rate of photosynthesis. So it doesn't matter how much carbon dioxide you add, it's going to be limited by other things. And those limiting factors will be light intensity, the amount of light, temperature, and the amount of chlorophyll. So common exam questions will ask, why is the graph plateauing, or explain the graph, and you would need to say that it's limited by other factors. And you can name one of the other factors. So if you pause the video, and if we can get that carbon dioxide is a limiting factor of the rate of photosynthesis, and also a sketch of the graph just to show what is happening, and you can annotate it with Y. Okay, next limiting factor is temperature. Now, temperature, like in chemistry where you did the rates of reaction, as you increase the temperature, the rate of reaction will speed up. And that's because those enzymes that control the reaction will be able to move around more and create more of the products. As we know, though, from the enzyme topic that we studied in organisation, that if the temperature is too high, that active site on the enzyme changes shape, and that means it can't do its job anymore. So we say that the enzyme has been denatured. So it will decrease and it will stop. So temperature, along with carbon dioxide, is also a limiting factor of photosynthesis. Now the graph for temperature is different to the other graphs. So we did carbon dioxide where it went up and then plateaued. In the temperature graph, it goes up. So as you increase the temperature, the rate of reaction increases until it reaches its optimum temperature, and then it very quickly decreases. And that's because the enzymes have been denatured. So if you can add to your revision about temperature being a limiting factor, Y is a limiting factor, and a sketch of a graph to show what the effect of temperature is on the rate of photosynthesis. Okay, so the next limiting factor that we need to know about is light intensity. Try not to say amount of light, use light intensity. Now, light intensity decreases as the distance between the plant and a light source increases. And that's important in the required practical, where you have to change a factor to measure the rate of photosynthesis. So you move a lamp away from a plant, and that will decrease the light intensity. Now, as you increase the light intensity, like with carbon dioxide, the rate of photosynthesis will increase. But again, like in carbon dioxide, you'll see that it levels off or plateaus. And that's because it's limited by other factors. So in this case, it will be limited by carbon dioxide concentration, temperature, and the amount of chlorophyll. So if you can add to your notes about how light is a limiting factor of photosynthesis, or light intensity rather, why it levels off on the graph. Make sure you've got a sketch of the graph as well. They do come up quite often in the exams. Okay. So in this section, there is the required practical, which you saw the video for. And one way in which we can measure the rate of photosynthesis is to use an aquatic plant, so a water plant like pondweed, and we can count the number of bubbles that it produces. And we presume that the number of that in the bubbles is going to be oxygen. Now, on the left-hand side, there is an example of a method that you could use to carry out this practical. So you can fill a boiling tube with 0.2% sodium hydrogen carbonate solution. Now, the reason we add sodium hydrogen carbonate solution is because that it provides carbon dioxide for the plant to make sure that carbon dioxide is not a limiting factor. We then add to the boiling point a length of pondweed, and we can cut the end at the top, and that just makes it easier to see the bubbles. We'll then add a lamp, and an LED lamp is preferable because it doesn't produce heat, which would affect the reaction. And we put it about 10 centimeters away from the boiling tube and let it to settle for a set amount of time. 5 minutes is normally a good amount. Once that 5 minutes is over, we can start the stopwatch and count the number of bubbles that it produces in 1 minute. Obviously, we would repeat it 2 more times and then calculate the mean number of bubbles. We'd then repeat the whole thing, doing it at 20, 30, 40, 50 centimeters away from the boiling tube. And that would change the light intensity, so we'd be able to see the effect. So what I suggest you do on this one is to put down some of the main steps of the method and why it is that we're doing certain things. That is a common exam question, to ask why is it that sodium hydrogen carbonate solution was used, or why is it that the pondweed is left to settle, or what would be a better lamp to use. You would put LED because it doesn't produce heat, and those kinds of things. So if you pause the video and put the required practical for photosynthesis, looking at the effects of light intensity on the rate of photosynthesis. Okay, next up. So we've spoken about all the limiting factors apart from chlorophyll. Now, chlorophyll is a pigment that is found inside the chloroplasts, and it's the pigment, the chlorophyll, that absorbs the light. So it's very, very important, obviously, in the rate of photosynthesis. Now, the energy that is absorbed from the light is used to convert carbon dioxide and water into glucose and oxygen. Chlorophyll is essential to that process. If there is a reduction in the amount of chlorophyll, so if there's less chlorophyll available, then the amount of glucose made will reduce, and the plant won't grow as well. Now, you can buy special plants called variegated leaves, and they have got white bits on those leaves. Now, they're often bred selectively to give those nice colours, so it looks nice in your garden, but the white areas don't have chlorophyll. Chlorophyll is green, so if you see green, that's the chlorophyll. So if it's got all these white bits on it, that's not able to photosynthesize, so they're not able to make as much glucose as if they were all green. So can we pop down how chlorophyll is a limiting factor in the rate of photosynthesis, please? Okay, so the next bit is we've made all this glucose from our photosynthesis reaction, and we need to know some of the ways in which glucose is used by the plants. Plants make glucose, but they need lots of different molecules, just like us. So they need proteins, they need fats and oils, they need storage molecules, they need cellulose for their cell walls, and they also use the glucose for respiration to get energy. So I'll go through the 5 main uses of glucose that we need to know about, and then you can add that to your notes. So glucose can be converted into amino acids, which come together to make proteins. Now, the way that they do that is they add nitrate ions, which come from the soil, to glucose, and that makes amino acids. Those amino acids then come together to make a protein. That's why it's really important that plants have got nitrates in the soil. So quite often people will feed their plants, but they're not feeding them because they don't eat food, but they are adding nutrients like nitrates to the soil so that the plant is able to make proteins. Another use of glucose is to produce fat or oils, which they use for storage. Now, quite often the oils that we get come from plants, and there will be lots in things like seeds and nuts, because they then have a store of energy ready for the seed to germinate. Another use of glucose, which is soluble, is you can convert it into something that is not soluble or insoluble. And the storage molecule that plants use is starch. We do not have starch or make starch. Plants do. It is their storage molecules. Quite often we eat the starch because it's a good source of glucose. So things like potatoes and carrots and turnips and things like that, what we're eating is just the store of glucose of the plant. Another use of glucose is to join it all together to make cellulose. And cellulose is really important because it strengthens plant cell walls. They do not have a skeleton or anything like that, so they need that strength in the cells so that they can stand upright. And another use of glucose, which comes up in the next chapter that we'll do tomorrow, is they use it for respiration. So plants respire as well as photosynthesize. So they can take that glucose, react it with oxygen, and they make carbon dioxide and water. So if you could pause the video now and add the 5 main uses of glucose by a plant. Okay, so that is it for the photosynthesis. So we're going to now do some questions, some quick questions, just to recall that knowledge and see if we can apply it in a different situation. So question 1 says, write down the word equation for photosynthesis. Make sure all your notes are covered, just in case you haven't done that, and do this on something fresh so that you can't see back. We're trying to do it from memory. So write down the word equation for photosynthesis. Question number 2. You don't need to copy out the table. You can just write water and then your answer, oxygen, your answer, etc. But can you put the chemical symbols for the molecules water, oxygen, carbon dioxide, and glucose? Question 3. Photosynthesis is affected by limiting factors. What is meant by the term limiting factor? And question 4. Name the raw materials needed by a plant for photosynthesis. So you might want to pause this video now just to give you time to answer all those questions. Okay, moving on to the next set of questions. Question number 5. Name the green pigment present in plant cells. Question 6. What is the role of this green pigment? Question number 7. For the graphs below, identify what the limiting factors might be in the experiments. So we have graph A, which has the x-axis labelled carbon dioxide concentration, and graph B that has temperature as the x-axis. And question 8, sketch a line onto graph A showing what might occur if the experiment was repeated at a lower light intensity. Slightly harder question. So have a go at what you think. Okay, we're going to move on to the last couple of questions. The last question, number 9. How is the glucose produced by photosynthesis used in plants? So if you could list as many of those 5 uses of glucose in plants. Okay, so we're going to move on. If you need more time, pause the video. And we're going to go through the answers to those questions. So question number 1, write down the word equation for photosynthesis. It's carbon dioxide plus water makes with light, oxygen and glucose. The chemical symbols for water is H2O. Please make sure that your numbers, by the way, are at the bottom, subscript, not at the top, so it's not H squared O. Oxygen is O2. Carbon dioxide is CO2, and glucose is C6H12O6. Question number 3, slightly trickier. Photosynthesis is affected by limiting factors. What is meant by the term limiting factor? It's a factor which is not at an optimum level to enable maximum rate of photosynthesis. So in other words, a limiting factor are factors that reduce or plateau or keep level the rate of photosynthesis after its optimum. Question number 4, name the raw materials needed by a plant for photosynthesis. They need carbon dioxide and water. Carbon dioxide they get from the atmosphere, water they get from the soil. Question number 5, name the green pigment present in plant cells. It's chlorophyll. Question 6, what is the role of this green pigment? So it absorbs energy and uses it to synthesize glucose or make glucose from carbon dioxide and water. Question 7, for the graphs below, identify what the limiting factor or factors might be in each experiment. So for graph A, the limiting factors will be everything other than carbon dioxide. So you could have put temperature and or light intensity and or the amount of chlorophyll. For the temperature one, the limiting factor is the number of active sites on the enzymes because they're being denatured. So if you've mentioned anything about the enzymes being denatured, then that will be your answer. Okay, last 2 questions then. Question number 8, sketch a line onto graph A showing what might occur if the experiment was repeated at a lower light intensity. Now you should have a line that looks pretty much the same as the original line, but it will be lower down. So it will still start at 0, 0. It will increase, but it will be at a lower level than the original line. And it will still plateau, but it will be lower than the original line. So pretty much the same, just lower down. Question number 9, how is the glucose produced by photosynthesis used in the plants? So there's 5 that we need to remember. So hopefully we got 5 or near enough to 5. So we got respiration to make fats or oils, to make amino acids, or you could have said proteins, to make insoluble starch, or just starch, and used to make cellulose for cell walls. So that is it from me today. Very well done for covering a whole chapter on that one. Hopefully you've got a nice set of notes. I know that I said in previous ones, but do make sure that you keep hold of these ready for year 11, stash them away somewhere safe so that you can use them for your revision before your exams. Tomorrow we're going to be looking at respiration, the types of respiration, the effects of exercise and what metabolism is, and then we'll move on to some exam style questions. So I hope you found that helpful and hopefully you've got a good set of notes in front of you. You will hear from me tomorrow for the next lesson. In the meantime, take care of yourselves and goodbye.